Welcome to this presentation. So in this video we are going to look at biology paper 2 prediction for 2026 GCE. So I have section A as well as section B. So for section A, the topics that you are likely to find in an exam, the first one is cell specialization. So what should you know about cell specialization? So these are just examples of specialized cells in plants and as well as in animals. So for these two here, they are coming from plants, and then this one here is coming from animal. So let's say they bring something like this one here, or you know to say that this is the ciliated cell, isn't it? It's a ciliated cell or a ciliated tissue, since it is made up of different ciliated cells. So a single cell of ciliated is called a ciliated cell. Then a group of ciliated cells is called a ciliated tissue. So this one is a ciliated tissue. So you know these parts of a ciliated tissue. So we have the cilia, which looks like air-like structure. We also have other structures like cytoplasm, nucleus, as well as the goblet layer here, as well as the mitochondria, which provide energy. So what should you know about this one here? Also have this one, where we have the xylem, which only transport water as well as mineral salts. So you have seen here, this is the function of the xylem, transporting water as well as mineral salts, as well as providing the mechanical support to the plant. So the way of flow, it is one-way flow. Okay, it's a one-way flow of sap. Then when it comes to phloem, actually this is not even the way the phloem looks like, but there are just other structures which are missing here. Like we can talk of the companion cells. So here, as you can see, this one is transporting water, sucrose, as well as amino acids. So these are the substances which are transported by the phloem. On this one, you expect it to transport in two-way flow of sap, so it can transport both up and down. So here you need to know the type of the cell. So the name, you need to know the name of the cell, when it comes to cell specialization, name of cell. Then apart from name of cell or name of the cell, just write it nicely here. So you have to know the name of the cell. Then apart from the name of the cell, you have to know the location where the specific specialized cell that they are going to bring in an exam is found. For example, this one is found in the lining of the respiratory tract, as well as in the lining of the oviduct or fallopian tube. Then these two, they are found in the vesicular bundles. So apart from knowing the location, you have to know the function. Function, very, very important. The function. So like I said, I've talked about the functions of these. Then the function of this just depends on the location. If it is in the respiratory tract, it sweeps out mucus containing dust particles. Then if it is found in the fallopian tube, it is going to move a fertilized egg toward the uterus, or just an egg toward the uterus. In cases of implantation. Then another thing that you need to know here, you need to know the adaptation. So I'm going to explain what adaptation is. So adaptation, you're looking at now the structures which are helping now the certain specialized cell to conduct or to perform the specific function. Like for example, we can talk of this ciliated tissue. It has these cilia which are air-like structure. So these cilia are the ones which are helping this ciliated tissue to perform its function, because as they are moving, they are sweeping out something, whether an egg or the mucus containing dusty particles. Okay, then apart from that one, you need also to know examples of other specialized cells in plants, in both plants and animals. So know examples of specialized cells in both plants and animals. So example of specialized cells in both plants and animals. Specialized cells in both plants and animals. So that is one thing that you need to know about specialized cells. Apart from that one, I can also include labels, because they can ask you to label this one, they can also ask you to label this one. So all those things, you have to know the parts. Okay, so I think on on the adaptation, you have heard what I was saying. You need to look at the structures which are enabling that cell to perform its function. For example, in talk of red blood cell, it lacks a nucleus, it is biconcave in shape. That's the reason why it performs those functions of carrying oxygen around the body or to tissues from the lungs, as well as a small amount of carbon dioxide from the tissues to the lungs. Then the second topic is enzymes. So what should you know about enzymes? So under enzymes, you need to know metabolism. Okay, you need to know metabolism. So you need to know that metabolism is divided into catabolism as well as anabolism. So we can have anabolism where we are building up small building blocks to form big blocks. Then also have catabolism where you can break down large molecules to form small molecules. And also you need to know examples of these reactions and where they occur in the body. Like for example, anabolism, you're converting glucose into glycogen. Where is it happening? It's happening in the liver. So that one is anabolism. Let's say for example, you talk of catabolism. Catabolism, let's say you're breaking down the carbohydrates into glucose. That one is a catabolism. Apart from that one, you need also to know the definition of an enzyme, being a biological catalyst. You need to know the function of the catalyst, that speed up of a chemical reaction, and characteristics of enzymes. Like these enzymes, they are made up of proteins and they are denatured at high temperature. They are denatured at different enzymes operate at different pH. Those things you need to know. Then the most important thing is this one, the enzymatic reaction. The enzymatic reaction. So as you can see here, you have to tell if this reaction here, let me just even do this. If this reaction here is anabolism or catabolism, you have to tell. Okay, you have to tell if it is anabolism or catabolism. So according to what I can see here, we have two substrates, two smaller substrates after the reaction here. At this stage of B, they are forming enzyme substrate complex. So this is called enzyme substrate complex. So you have to know the stages. This one is a substrate, or this is a substrate, this is an enzyme, and then this is called enzyme substrate complex. So you have to know that one also. It's very, very important. Complex. And then after that, as you can see here, the product is being formed. It's a bigger one. It's different from this. So meaning to say this reaction is anabolism. And there are some situations whereby you start with this one and then you end with this one. That reaction is catabolism. So you have to know that. Apart from that one, know the industrial application of enzymes. It's very, very important, like in the use of the enzymes in blowering, the use of enzymes in leather tanning, as well as use of enzymes in different aspects. And then also have this part here, one of the factors that affect the rate of enzymes is pH. So we have different enzymes, enzyme 1, enzyme 2, enzyme 3. So this enzyme here is operating at acidic because the pH is low. So when the pH is low, otherwise the pH scale ranges from 0 to 14. Okay, so the pH at 7 is neutral, then the pH from 0 to 7 is acidic, then from 7 to 14 it is basic or alkalinity pH. So when an enzyme is operating under acidic, meaning to say it can't operate under alkalinity. And that is one thing that you need to know. Different enzymes operate at different pH. So this enzyme 1 is operating under acidic, this enzyme 2 is operating at slightly acidic, slightly basic, because here at this point here it is in acidic, then it is entering the alkalinity or basic pH. Then for this one, enzyme 3, it is actually at alkalinity or high alkalinity. So you have to know examples of enzymes. For example, in talk of enzyme 1, it can be pepsin, because we know to say that pepsin, it is produced in the stomach, where the stomach produces hydrochloric acid, and hydrochloric acid is an example of an acid. So that is where it works well. On this part here, enzyme 2 can be the duodenum, because we know, let me say, it can operate in the duodenum, because we know to say that the content that comes from the stomach, it enters the duodenum before being converted into alkalinity. It is actually acidic. So once the bicarbonates are being produced, that's when it is going to be converted into what? Into alkalinity or basic. So example of those enzymes, you can talk of pancreatic amylase. Apart from pancreatic amylase, you can talk of trypsin, all those enzymes. Then for enzyme 3, you can actually talk of amylase, okay, or salivary amylase, I can say. Salivary amylase, the mouth is alkalinity, pure alkalinity, unless there are some conditions whereby someone is having the disorders. That's when the mouth is going to be acidic, but actually it is alkalinity. So those things that you need to know. Apart from that one, temperature, the effect of temperature and other things. Let's see the other topics that you need to understand. So we have nutrition and digestion. So under nutrition, you need to know the types of nutrition. You need to know digestion as well. So under type of nutrition, of what we call saprophytic nutrition. Saprophytic nutrition. So under saprophytic nutrition, we have what we call a lysopause. So this one is a lysopause. Let me say lysopause. So under lysopause, you have to know the parts of the lysopause as well as the function. So the entire part here is called the sporangulum. Then these tiny balls, they are called, these tiny ball-like structure, they are called the spokes or the spores. So you have to know the spores, the balls of the spores as well as the function of the spores. Then we have what we call the stolon. So stolon is more like a connector, the one which connects two different lysopause. Okay, yeah. So actually lysopause is this one, not like that one. Yeah, the correct spelling is this one. So when it comes to the structures, also have the stolon, the one which anchors. It's more like the roots. It acts as a root and it anchors to the substrate, and the substrate can be the dead or the decay matter. It can be the decaying matter or it can be the bread. Okay, so these lysopause, they can even germinate on the bread, because this one here, it releases enzymes that hydrolyze starch into maltose. So maltose is the one that this lysopause uses. So you have to explain how this one here absorbs nutrients from the substrate, for example the bread or the dead or the decay matter. You have to know the functions. So the functions of the lysopause, or the lysoid, I mean, this lysoid secrete one enzymes. Apart from enzymes, it also secretes it also anchors the lysopause to the substrate. So you have to know that one. Then let's come to the digestion. You have to know the parts, the mouth, the esophagus, as well as the function. So this is where the peristalsis or peristalsis occurs. The stomach, the function of the stomach, then the liver, the metabolic function of the liver, as well as the ailments of the liver, like liver cirrhosis, hepatomegaly, hepatitis, all those things. We also have the gallbladder, the function of the gallbladder, like storing bile, the function of the pancreas in terms of exocrine as well as endocrine, releasing hormone as well as releasing enzymes. The function of the small intestine, site for the absorption of nutrients, different nutrients. Then for the large intestine or the colon, it can be the large intestine or the colon. a site for the absorption of water and electrolytes. So this is the rectum and this is the anus. So the rectum, this one just store feces temporarily, or store feces. Then in terms of they can ask you where the digestion of certain nutrients happen, like for example the digestion of proteins, and also where different enzymes are released. For example, if they ask you where pepsin is released, it's released in the stomach. If they ask you to say where lipase is released, it's also released in the pancreas as well as in the stomach. So those things are the ones that you need to know. Even the stomach, even the small intestine releases enzymes, which are the brush border enzymes. So you have to take note of that. Then we have the second one, which is about the movement of substance across the membrane. So we have the animal as well as the plant. So you have to know the types of solutions and what will happen when you place a certain cell, for example an animal cell, which is a red blood cell, in hypertonic solution, as well as isotonic solution. So when you place it in hypertonic solution, it is going to lose water and shrink by osmosis. Then when you place it in hypotonic solution, it is going to gain water by osmosis. So you have to explain the mechanism, not the way I'm explaining. You have to say, when a red blood cell is placed in hypotonic solution, it will gain water by osmosis, since the concentration inside the red blood cell is greater than the concentration outside the red blood cell. Therefore, water is going to move from outside the cell into the cell, or into inside of the cell. The cell will gain water by osmosis in that way. Isotonic, iso means same, hypo means low, hyper means high. So isotonic, the concentration is the same, the one which is outside and the one which is inside. So the cell is just going to maintain its shape. So in this scenario, you find that the cell is going to burst if it is an animal cell, because it doesn't have the cell wall. And then when it comes to the plants, you have to know the movement of substances, like for example water from the ground to the leaves. So we have the root air cell, we have the root cortex, we have the xylem, the one which transport water, and you remember transport on one direction. Let's come to the plant. So isotonic, hypotonic, hypertonic. So when it is placed in isotonic, nothing is going to happen. There is no net movement here. When it comes to this one, hypotonic solution, when you place a plant, it will gain water, but it won't burst because of the cell wall. Then when you place a plant cell in hypertonic, it will lose water, and it is going to undergo plasmolysis. So this one is called a plasmolysed cell. So all those things you have to know them. But from that one, you also need to know diffusion. You have to know diffusion and examples of diffusion. Okay, like oxygen diffuse out of the plant, of the leaf, using the stomata pores. Even carbon dioxide diffuse into the leaf using the stomata. Even oxygen diffuse into the lungs using the alveolar. All those things, and carbon dioxide and other gases, in short. Let's see the next topic. So the next topic is genetics. Actually, this one here has been already done for you. So what it is here, we have the antigen or antigens on the red blood cell. So another function of red blood cell is that on the surface membrane of red blood cells, that is where you are going to find the antigens. So these antigens are very, very important in determining the blood group or the blood type of someone. Then we have the antibodies. So antibodies, they are derivatives of the antigens. So antigens are similar to the blood group. So if someone is having blood group A, even the antigen is going to be A. Someone is having blood group B, the antigen is going to be blood group B. But for O, it doesn't have antigens, but it has the antibodies. And this is the reason why group O is the universal donor and the blood group B is a universal recipient, because blood group B doesn't have the antibodies. So in terms of the serum, In the serum, you are going to find the antibody B, then antibody A, then antibody A and B for blood group O. So this is very important, and you have seen the way I'm writing these, or the way they have written these. I can say they are in small letters, so you have to say anti A, anti B, anti A, and anti B for blood group O. So apart from that, you have to know the terms which are used under genetics. Know the terms like the allele. You have to know the definition of an allele. You have to know the definition of the genotype. You have to know the definition of phenotype. Okay, so phenotype. Let me just write it nicely. So you have to know the definition of the word phenotype. Okay, phenotype, as well as other terms like inheritance, a gene. You have to know the definition of a gene. You have to know the definition of homozygous dominant, heterozygous dominant, codominance. All those things are very, very important. And nevertheless, you have to know how to draw the genetic diagram, which is very, very important. So under genetic diagram, you have to know how to find the probability. You have to know how to find the probability. This is very, very important. And then apart from that one, you need also to know sex-linked disorders. Sex-linked disorders, the definition of the sex-linked disorder or characteristic. Then apart from that one, you have to know how to draw the genetic diagram using the idea of sex-linked disorder according to the question that they are going to bring. You have to know mutation, definition of mutation, types of mutation, and substances that causes mutation. You have to know all those things and the effect of mutation, as well as variation, examples of variation in plants and animals according to discontinuous as well as continuous variation. So you have to know that. Even the definition of variation is very, very important to know. Then another one is excretion. So under excretion, you have to know the nephrone, actually the kidney, the parts of the kidney, and the functional unit of the kidney, which is the nephrone. So under the nephrone, you have to know the types. Let me say the parts, even the types of nephrones. Now at your level, it's not even very important, like medullary nephron as well as cortical nephron, or juxtamedullary nephron and cortical nephrons. Those are not even very important. So the parts, if the blood is coming from this side here, it enters the glomerular, then it is going to leave the glomerular using this blood vessel. So you have to know these blood vessels. Like this part here is called the afferent arteriole. Afferent arteriole. The afferent arteriole. Not like this. It's not the way we write arteriole. Let me just write it nicely. Arteriole. The afferent arteriole. And this one is called the efferent arteriole, or efferent end, if you want. This is the efferent end, and this is the afferent end. So you have to know this. Then also have this part here, which is called the proximal convoluted tubule. We also have distal convoluted tubule. So you have to know what will happens at distal convoluted tubule. Actually, just the absorption as well as secretion. So this is where the absorption of water as well as glucose takes place. So from there, you have to know the substances which are filtered out here at the glomerulus filtrate. Them is at the glomerulus. So this is the glomerulus, and this cup-like structure, this one which I'm drawing, the space here is called the Bowman's capsule. This one is called the Bowman's capsule. The Bowman's capsule. Okay, so the substances are going to be filtered into the Bowman's capsule. They are going to travel using this tube here, which is called the proximal convoluted tubule. So in the proximal convoluted tubule, that is where now the absorption of glucose takes place as well as water, but not all amount of water is going to be absorbed. But for glucose, it's 100% the absorption. That is where it is. it is going to be reabsorbed. We also have other parts like the thin descending loop of N, but actually the entire part is called the loop of N or the loop of N. Now this loop of N, it has parts like a thick as well as, let me say, the thin as well as the thick. So we have the thin descending and thick ascending as well as thin ascending. So you have to know the substances which are reabsorbed here. So have these substances like salts, the absorption by diffusion. Also have on this part here. But when it comes to thick ascending, only solids are reabsorbed, not water. Also have the distal convoluted tubule, which become permeable under the influence of antidiuretic hormone. When the person is dehydrated, even the collecting ducts, they become permeable to water when ADH is released, antidiuretic hormones or hormone. So you have to know those things. Also examples of excretory organs like the kidney, the skin, the lungs, and other things you have to know. And also the substances which are secreted by those excretory organs. Then let's come to section B. For section B, as you all know, to say that section B is the essay part. So you have to write an essay on this part. So one of the topics which one of the topics or the topic which is likely to come is excretion. So you have to describe the process of excretion, how it is carried out using an example like the skin. Also you have to explain the importance of excretion to animals as well as for plants. Okay, so for the part of the kidney, you have to identify the internal structure of the kidney. So they can just bring the structure of the kidney. Then you so let's say, for example, if this is the kidney. So if this is the kidney, you have to label the kidney, the parts of the kidney. Okay, so let me just draw a rough sketch of the kidney. So let's say, for example, this is the kidney, a rough sketch of the kidney. So you have to label the structures. Like for example, this is the cortex or the renal cortex, and this is the medullary, which is also known as the pyramid. So this is the pyramid, and the entire structure is the medullary. Then we have the renal pelvis, which is this part here. We have the ureters, we have the renal artery, we have the renal vein. All those structures you have to label them and you have to know them. Sometimes when they ask you to say explain the mechanism of excretion in the kidney, you have to draw the nephron. Then you explain. After drawing the nephron, you label it. You explain the mechanisms of filtration, the substances which are filtered on the glomerulus and the substances which are not filtered on the glomerulus, and the reasons why they are not filtered. Like blood cells are not filtered because they are big molecules. Proteins are not filtered, but glucose is filtered. But you don't have to say that glucose is important, so it is going to be absorbed in the proximal convoluted tubule. So when you use term proximal, the word proximal means near to something. So this proximal, it is near to the glomerulus or the Bowman's capsule. Then distal means away from something. Okay, so that is one thing that you need to know. Then another one, you have to identify common disorders and diseases associated with the kidney, like kidney failure, kidney stones, all those things. Then also the application of the dialysis machine as well as kidney transplant, because they can ask you to say what are the ways of managing disorders of the kidney, like using the dialysis machine as well as undergoing kidney transplant. All those things you have to know them. In terms of the lungs, you have to investigate the role of lungs in excretion. So on this one, if you want, you can draw the lungs when the question is actually carrying a lot of marks, and then you explain and then you show. But actually you have to draw the alveolar. So let's say, for example, this is the alveolar. You just need to draw a simple diagram of an alveolar. Then you label the substances which are entering and the substances which are leaving the alveolar. So let's say this is oxygen and this is carbon dioxide. Then you also draw a the blood capillary here like this. You draw it simple. You also say this is diffusion, these they are entering by diffusion. So you give examples of substances which diffuse. Okay, the law of this one is in excretion. So you are going to focus much on carbon dioxide, the one which is excreted, as well as heat. So heat is excreted from the lungs in form of water vapor. Yeah, so you have to take note of that. Carbon dioxide as well as heat. Then the second one, under the human skin, you have to identify the structure of the human skin. So if they say explain the role of the skin in excretion, you have to draw the skin. You label the parts, then you start now explaining. Sometimes they can say explain the functions of the parts of the skin. You have to draw the skin. But how do you start an essay? So I've created a video on how to write an essay. So feel free to watch that video on my channel. Then another one, you have to describe the role of human skin in excretion, which I've already talked about. And then here we have homeostasis as well as osmoregulation. So you have to know the definition of homeostasis as the maintenance of the constant internal environment. So this is very, very important. For example, if your blood sugar is high, this one is going to make sure that your blood sugar is on the normal level. Even the temperature, when the temperature is high, this one is going to make sure that your temperature is on the normal level, like 35 to 37 degrees Celsius. So you have to take note of that. Then you have to identify important organs in homeostasis, like the liver, the function of the liver in homeostasis, the function of the kidney in homeostasis, like acid-base balance, as well as the lungs, homeostasis, the skin, as well as osmoregulation. You have to describe all osmoregulation of the skin, all those things. So the next one is saying describe the role of the kidney in maintaining the balance of water and salt ions. So this is very, very important. You have to draw the kidney or the nephron. You explain. So for this part, you have to involve the ADH. Okay, so the ADH, which is antidiuretic hormone, the one which is released when someone is dehydrated to cause the absorption of water in the nephron of the kidney. So here, let me just bring you back. So here you have to describe the mechanism of thermoregulation by the skin when it is cold, when it is hot. How does the skin respond in terms of the blood vessels? Are they going to vasoconstrict or vasodilate? In terms of the follicle air, are they going to stand or are they going to constrict? Okay, or dilate. Also have this part here which is saying describe the role of the liver in regulation of blood sugar and body temperature. So the liver, it has one of the metabolic functions of the liver is heat production. So I have to explain that one. And also blood glucose, that is converting glucose into glycogen, as well as converting glycogen into glucose. When there is insufficient, when there is low glucose, it is going to convert glycogen into glucose. When there is high glucose, it is going to convert glucose into glycogen for storage. Then when it comes to the hormones, you have to know the definition of hormones. So if they say describe or what is meant by the term hormone, 3 marks, you have to explain as a chemical substance which is secreted by ductus gland, which travel using blood, and it has effect on target organs. So those things. And also you have to identify the endocrine glands in human, like pituitary gland, pineal gland, you have to know thyroid gland, and also the hormones which are secreted by those ducted glands, like talk of the pancreas, it secretes insulin as well as glucagon. Talk of the kidney, on top of the kidney we have the adrenal line. Also the kidney secretes hormones like thrombopoietin, all those learning, all those things you have to know. Then under skeleton and locomotion, you have to identify various types of skeletons, like hydrostatic skeleton. We also have exoskeleton. We also have endoskeleton. You have to describe the function of the skeleton. If this one comes carrying 6 marks, saying describe the function of the skeleton, just very simple: protection, provide mechanism, or provide the attachment for the muscles, and other things. Then we have this one, which is saying investigate the structure and composition of exoskeleton, identify joints and muscles in the limb of the grasshopper, identify the bones of the exoskeleton as well as appendicular skeleton. So in terms of the exoskeleton, it consists of the skull. It's made up of cranium, just the upper limb. Then for the appendicular skeleton, consists of the girdles, like the pelvic girdle, the one which also connects to the trunk, which is the thorax. And then explain a bone as a living tissue. So this is very important. It can carry 4 marks or 2 marks. It contains white blood cells or blood cells. It can be affected, as well as it contains red blood cell, white blood cell. It can get affected. In other words, it can be repaired. That's the reason why a bone is considered as living tissue. Then muscles and joints. You have to describe the structure of the skeletal muscle. So here you have to draw the structure of the skeletal muscle as well. Then describe it. You have to describe the function, okay, in terms of contraction and relaxation, in that way. And then under the reproduction, you have to describe sexual reproduction. You have to describe the differences between sexual and asexual reproduction. You have to identify male and female reproductive organs in human beings. They can ask you to say explain the functions of the male reproductive organ, or the different parts of the male reproductive organs, the function of the different parts of the male reproductive organ. So you have to draw a male reproductive organ and you start explaining their functions. Then explain the function of the different organs of the male reproductive system, which I've already explained. Then this one here, which is the last one, you have to describe the biological changes associated with sexual development in human beings, like for females, as well as causes of infertility in humans, like consumption of use of contraceptive pills, hormonal imbalance, abuse or substance abuse, like use of marijuana and other things. So just that we are remaining a few days. Otherwise, for those who want to register with me, you can just contact me or WhatsApp me on 0968 01 47 43. Only for serious business, 100 kwacha per subject per month. Doesn't matter whether you are remaining with a few days to write. The matter is that you have to pay 100 kwacha per month. That's all. So for section B, you have to use the syllabus. So the syllabus or the syllabi, you have to download it on Google. Just say biology 10 or grade 10 to 12 or Zambian biology from grade 10 to 12 PDF. Then you're able to download that one. Thank you very much for watching, and see you next time in the next video.